Hello my dear students, I hope all of you are doing very well. My name is Abhijit Devnath. I am working as an assistant professor at NIT Pharmacy Institute. So, I am teaching you this subject called Pharmaceutical Biotechnology, and we are actually learning the unit 2 of Pharmaceutical Biotechnology in your this BPharm course. So, we have learned a lot of things in our previous lecture. We also have seen that how all these new technologies in biotech is helping us to solve our current days major challenges in the healthcare sector. We have seen about the rDNA, recombinant DNA technology, how genetic engineering field is very that is nicely getting diverse and solving the current days problems like vaccine production, antibody production, monoclonal antibody synthesis. So, today I am going to tell you a very interesting story, a very interesting concept about the hepatitis B vaccine. I hope many of you might have taken hepatitis B vaccine during your childhood days. So, today we will learn how you will be able to produce hepatitis B vaccine. Before that, I will start my lecture with telling you what is hepatitis B. I am going to tell you about the vaccine. I am going to tell you how you can produce and about the various marketed formulation. So, let me start with you hepatitis B. So, you can see this is a very bad thing, this is our enemy, we do not like these things, we hate these things. You know why? Because this is hepatitis B, hepatitis virus, and you know what, this virus is actually attacking our liver, this virus is attacking or destroying our liver cells. Can you imagine? And you know why liver cells are important? Liver cells are actually very much important for us, because due to liver, whatever the things we actually consume, whatever the food, drugs, other substances, whatever the things we actually consume, the big molecules, they actually get metabolized by liver. I hope all of you might have studied about there is called phase 1 metabolism, then phase 2 metabolism. I am talking in the context of drug. If you take some drug, it will, if you take some drug orally, it will ultimately reach to your liver. There will be two phases of metabolism, phase 1 and phase 2. Now, there are lots of cells, liver cells are actually present there. These things are actually helping in metabolism. Now, can you imagine if your liver get destroyed, if your liver is not working properly, then what will happen? Obviously, metabolism will not happen. If metabolism will not happen, your entire body will suffer, because all the poisonous things will be that is stored in your liver and other areas in your entire body circulation. If liver is get failed, you will not able to survive. You might have seen those people take alcohol very commonly, they take alcohol, their liver, they actually suffer from liver damage. Why? There are lots of reasons, I am not going to tell about that thing, but for you to understand, liver is very important organ in your body. Now, this virus, it is attacking our liver. So, hepatitis B, it is a disease caused by the hepatitis B virus, an envelope virus containing partially double stranded circular DNA genome, this virus. In our ancient, it is an ancient disease described in the 5th century BC. Can you imagine how long, how old this virus is? Earliest recognized blood-borne outbreak of hepatitis was in Germany in 1883, receiving smallpox vaccine. In 1947, McCollum and Weir introduced the term of hepatitis A for infectious and hepatitis B for serum hepatitis. Okay, they have in 1947, McCollum actually tried to separate. The terminology was adopted in 1973, and then the term hepatitis described the inflammation to the liver. Before that, it was not clear. Hepatitis may be caused by the alcohol, drugs, also autoimmune diseases and metabolic diseases and other viruses. Viral infection may account for the more than half of the cause of the acute hepatitis. See, if your that is our liver is already damaged, then obviously it will be not able to work properly, and then for virus it is a great opportunity to destroy. Now, how you can stop the virus, or how you will be able to that is save ourselves. here okay, zooming is not available. Anyway, I think the screen is not much more rightly visible. So, vaccine, how you can fight against this hepatitis? The best answer is, first answer is the drug. Just a second. The first answer is the drug. The second answer is the vaccine. See, if somebody's liver is already damaged, if somebody's liver is already damaged, at that scenario, do you think drug will properly get metabolized like phase 1 and phase 2? Answer is no. At that scenario, viruses are actually present in liver and other areas. What is the other option? Other option is triggering, activating our immune system. Our B cells, our T cells, macrophage and entire our immune system, if you may activate them, so that they can produce the antibody and your entire T cells can destroy those viruses, this will be a great option. That is why we have to work on vaccine. If I tell you the story of vaccine, you can see, I do not know the screen is how much visible to you or not. In 1970, entire the vaccination campaign was started. Here is a picture of 2010 up to 2010. So, what happened? In earlier time, at the beginning, smallpox vaccine was given, and after that plague, cholera, typhoid and rabies vaccine was also given here. It is still continuously given. Then toxide and other polysaccharide is actually given in the form for various diseases like influenza, yellow fever, diphtheria, tuberculosis, tetanus, pertussis. And after that polio, pneumococcus and other various kinds of virus bacteria, for other viruses vaccine was given. But for hepatitis B, it has taken almost long time, up to I think from 1880 to 1890. In that period, hepatitis vaccine was actually become so much popular, and in 1990 hepatitis A vaccine was also given to the patients. And then you have seen the other vaccines like typhoid, various kinds of typhoid, influenza, rotavirus, HPV, human papillomavirus, zoster and prostate cancer, other vaccines are given. Now, you may imagine how much late we are. We are much more late when we talk in the context of hepatitis B vaccine. Why, sir? Why you are saying so? Because our vaccination journey, complete vaccination journey was started almost you can say 1750, and when we have started our vaccination for hepatitis B and other things almost 1880 to 1890. Before that, whatever the population we have, those who have encountered with the virus, they already might have, that is, are not able to survive, because drugs, obviously we are using various kinds of antiviral therapy, but it is not easy. If somebody is fine, if somebody is very good, fit and fine, if you give the vaccine to that person, in his entire life, this person will be not affected by hepatitis B virus. Whatever the virus you have seen here, this virus will be not able to attack you. If you give the vaccine to a patient in his that is fit, when he is fit, before attacking any of the diseases, his immune system will be much more stronger, so that his immune system will attack if virus may enter. All the antibodies will be there, it will be do some neutralization or opsonization, whatever the procedure, then T cells will come, bind and destroy. Okay. So, after that we can say in 2010 we have seen prostate virus, prostate cancer and zoster HPV are giving, and recently we are actually developed the COVID vaccine. This is happening in 2021. Okay. So, you can imagine where our that is hepatitis B vaccine journey actually started. Now, if I tell about the hepatitis virus vaccines, types of vaccine, what kind of vaccines actually given. See, since 1970 to 1980, the plasma derivative actually is to given, plasma derivative. Scope, it was, yes, it was effective. What is the limitation? Safety limitation was there. Plasma derivative actually is to given. After 1970, after 1980, since 1980, recombinant DNA or recombinant vaccine was actually given. Why? These are the potentially safe and effective, and these are actually no, that is if I say non-responder, in many of the cases you will be observing during clinical trial non-responder. Many of the people, their immune system, their body will not respond against a certain that is a vaccine. So, number is very low, 5 to 10%. After that is a 2000, there are that is multiple surface antigen actually given. These are also safe and effective, cost, and its problem is the cost, high cost, and it has limited use. Present end, what we are doing? We are actually using use of second generation vaccine in the global context, explore the different types of not responder, why they are not responding, okay. Development of third generation vaccines, then development of other formulation vaccines with the neoadjuvant, exploration of the dose and route of administration. These are the things are going on in present day scenario. Ultimately, we are focusing here because it is potentially safe and effective. Yes, some people are not responding. So, we have to that is explore about the non-responder. If we make some genetic changes, then it is there are chances those responder non-responder people, their immune system will also work, they will also able to respond. So, that in their body some antibody will be created, and if the virus enter in their body in future, it will be able to destroy this virus. Now, how we are actually giving? So, you can see this is the infection pathogen. For example, this is the hepatitis B virus. So, you can generate the antibody to the patient's body by using this process, either inactivated, you may remove, you can take this virus, you can destroy its genomic material, or you can make it as an attenuated, okay, or you can further after attenuated you can other do some reassonant, some of the part will be not activated, or you can split, or after splitting you can give some subunit or irreversible to the patient's body. So, what will happen when any of these things, this is called as an antigen. Okay. Vaccines are basically antigens. When these things will enter in the human body, obviously, all of you have seen in my previous lecture, cytokine, interferon, this will be activated, these things will be informed to other cells, T cells, B cells will come to the picture, B cells will try to produce some antibody against those the particle, those substances. And there is something called memory B cell. Memory B cell will kept that antibody for long period of time. In future, if the real virus may enter in their body will be able to fight against that real virus and it will destroy. Okay, so, you can prepare hepatitis B vaccine from this way, and now if you make this way, so the protein antigens are actually produced. You can use the recombinant DNA technology. I am using, you can use this way, but for example, you are using the subunit or purified part or resonant part or inactivated. You have to make some changes of this virus. You should not give the actual virus. You have to make some changes to the virus, so that your body may think it is a foreign substance and your body may create some antibody. Now, to make the vaccine ready, you have to take the help of the rDNA, recombinant DNA technology, where the DNA sequence coding of the antigen protein is actually inserted into an expression system that is then able to produce large quantity of the specific antigen. For example, this panel A, where I have used the in vitro, panel B, this is for the using the DNA plasmid, and panel C, in the live vaccine or vector. See, this is a pathogen. If you clone the gene coding antigen into DNA plasmid, this will be called as a recombinant DNA. Or if you take in the live vaccine vector means live vaccine vector, you can see live vaccine vector, not live virus. Lentivirus or other kinds of retrovirus, those are not harmful to us. Viruses which is not harmful to us, you have taken that genome gene from the virus and you are actually trying to adding here, but you have removed all the bad genetic part. You have actually removed the bad genetic part and you have tried to add with this virus, this hepatitis B virus. Entire part you have not given, you have removed the bad or the dangerous part or the genetic code which is actually making the virus dangerous, harmful. Basically, you have tried to give the epitope area, spike or other epitope areas code. Okay. Then insert gene into the expression system, then protein expression will be happen, and then purification. After purification of rDNA, whatever the things will be getting, then it will be insert into the human body. Okay. These are the steps. So, the vaccine generated using the recombinant DNA technology is called as a recombinant vaccine, and these types of vaccines are manufactured with the assistance of expression systems such as bacteria, insects, yeast, plant and other things. This are learned. Usually E. coli expression is the workhorse and most popular organism used for recombinant protein expression, due to in part with its ease of bioprocessing, scale up and strain engineering, lower cost and shorter production of time. See, this process right now is much more very much easier, that is why people are preferring, and cost is very low, short production time is actually required. I have told you at the beginning of my the lecture series, a great scientist called Kate Broderick. She is I think working AstraZeneca after the outbreak of SARS-CoV-2, that is coronavirus. When China released the genomic sequence of SARS-CoV-2 in probably in NCBI, she has downloaded. After that, she has analyzed the sequence, entire sequence. She has tried to develop some mRNA based vaccine. She has taken only 15 minutes. She has taken only 15 minutes to make the entire mRNA vaccine, but she has done in computer. After that, she has done the work on in vitro and in vivo, in lab and also in animal. For that, it has taken much more longer time, but the designing part was only for 15 minutes. So, how she has done? There is an entire documentary is available on BBC. If you may search on Google, BBC Kate Broderick, coronavirus vaccine, mRNA vaccine, you will be getting the video. You will be able to see in her lab how she is working. You will be able to see such kind of round picture. It is actually plasmid in this part, in sorry, just a second. Yeah, in this part the gene of interest was actually added. So, that is why it is a very simple process and it is becoming so popular. Now, a combination of simple genetics, ease of culturing and scalability, fast expression and high yield make E. coli as a attractive choice. If I tell you more, there are various types of vaccines are made possible using rDNA. One of the process is the DNA vaccines, another is the recombinant protein subunit vaccines. In DNA vaccines, process again very simple. This is the genomic material or gene of interest. So, copy from the DNA of pathogen and this is the plasmid. Here is the empty plasmid, nothing is here. You are actually by using restriction and a nucleus, you are cutting this restriction and this plasmid, you are adding this genomic material, the gene of interest into the plasmid by using ligase, then plasmid is actually constructed and then whatever the DNA actually you have got here, you are giving directly to the human body. this will be called as a DNA vaccine. Okay. If you take RNA, in this case called RNA vaccine, but in case of recombinant protein vaccine, for example, here hepatitis B virus, either as DNA or RNA you may take. So, hepatitis B antigen producing vaccines gene, hepatitis B antigen producing gene you have taken, and on the other side you have taken one bacteria. You know, in that bacteria plasmid is present, you have taken that plasmid, you have cutted that plasmid by using restriction in nucleus, and you have inserted that part here. You have inserted that part here, and now you have tried to introduction of the recombinant DNA into yeast cell, yeast. Okay. When you will insert that thing into yeast cell, what will happen? It will further divide. Okay. So, you can see hepatitis B antigens are actually producing within this, and then after doing your recombinant yeast cell multiplying and producing this hepatitis B antigen in the fermentation tank, ultimately you have to do extraction and purification, and then you will be getting the hepatitis B vaccine. Okay. The process is very simple, very interesting. In future, I wish you will be able to working in some pharma company where they are doing such kind of things. So, the HB virus is produced by expressing hepatitis hepatitis B virus surface antigen. Here, whatever the gene of interest we have taken, this is the surface antigen. Okay, by using the yeast secretion. Yeast secreted the antigen into culture supernatant, facilitating the purification. Yeast post translation machinery is suitable for this purpose. It renders the antigen with the necessary glycolization pattern. Upon the recombination pattern, recombinant expression, the HBS, it is Hg, assemble into the virus like particle VLP, virus like particle. So, whatever the things it will produce na here, it will produce the virus like particle, which will be immunogenic and making the effective vaccine. Here is the entire process, whatever I have told you, you can see again. So, this is the vector virus. You can also sometime use the live recombinant vaccine development. Virus target, you have taken that part, you have inserted into the vector, and then you have done some large scale fermentation, and ultimately your commercial vaccine is available. Now, you can do the vaccine. These are the few example of marketed formulation. Can you see something special in this picture? There is something very important thing in this picture. What is these things? It is this thing, Bharat Biotech. Do you know, my dear students, when we got independence in 1947, our country was not so much developed in the field of science and technology, especially drug discovery and vaccine development. Those days we used to so much dependent on foreign companies. They used to send various vaccines, but this time in 21st century, what happened? One is Bharat Biotech and another is SII, Serum Institute of India. We are really proud on them. We are really proud on Serum Institute of India, then your Bharat Biotech and all the other biotech company, pharma biotech company, those who has developed the vaccine, and they are exporting Indian vaccines to the other countries. Right now, we are not exporting vaccines from other countries. Many of you might be telling me that, sir, we are importing Sputnik. Okay, that is okay, but right now we are capable to make our own vaccines in our own country. That is why we are really proud on these organizations like Bharat Biotech, Serum Institute of India. I have a great salute to all the scientists, those are working in this project. Now, why I am telling you all those things? Because our Indian companies, our Indian Bharat Biotech, they also developed one hepatitis B vaccine, which is a recombinant hepatitis B vaccine, okay, which is a recombinant hepatitis B vaccine. Here is the detailed that is whatever they have given in their website. I have taken from their website for this educational purposes. So, they have also mentioned that this vaccine, it is also endorsed by various reputed organization like American Academy of Pediatrics, Center of Disease Control Prevention in Atlanta, that is US, then European Medicines Agency for evaluation of the medicine for human use. Okay. This is the entire details whatever they have mentioned in their that is a prescription document. Yeah, you can see they have mentioned entire the recombinant DNA process, also whatever the qualitative etc., it will be available in the suspension form. Okay. So, we are very proud on Bharat Biotech, another nation also in Indian, those are actually producing vaccines. This is another marketed formulation, it was made by Mark and Sharp, and you can see these are few other marketed formulations which are also available in the market for hepatitis B vaccines. So, today we have learned these things, hepatitis B disease, its vaccine, how you can produce and about the marketed formulation. Next day I am going to tell you something more interesting thing, insulin. Yes, you can use insulin to fight against diabetes. Introduction to insulin, I will tell you insulin production by rDNA technology and about the various marketed formulation. So, thank you all of you, thank you for joining me, it is a great time to teach you all. We shall meet in our next lecture. All the best, God bless.